Hi, I'm Andrew Westbrook. I'm an assistant professor in psychiatry here at Rutgers and a member of the Center for Advanced Human Brain Imaging Research. In my work, I study the balance between excitation and inhibition in the brain and implications when this balance is lost for psychiatric and neurological disorders. Healthy adult brains contain both excitatory neurons, which increase neural activity, and inhibitory neurons, which decrease neural activity. And it's the balance between these two which give rise to special properties that make thinking possible. When imbalances occur, the consequences for brain health and for cognition can be dire. I study imbalances in excitation and inhibition in two very different disorders, in depression and in Alzheimer's disease. To study depression, I've teamed up with Dr. Cheryl Graber, who's a psychiatrist here at Rutgers, to understand how patients with major depression have differences in their brain excitability, but also how treatments like TMS or transcranial magnetic stimulation might be able to alter excitability in ways that could remediate symptoms. Our goal is to find ways to make treatments like TMS more effective. To study Alzheimer's disease, I've teamed up with Dr. Mikhail Beery, who's director of the Herbert and Jacqueline Krieger-Klein Alzheimer's Research Center here at Rutgers, to understand how changes in EI balance or excitation inhibition balance can be measured using tools like electroencephalography or EEG. We're testing whether we can detect these changes in brain activity early before symptoms start. Our goal is to understand both how the disease progresses, but also to find early warning signs so that we can intervene early and alter disease progression. To study these disorders, I work with both TMS and EEG, which is part of a broad suite of tools we have here at the Center for Advanced Human Brain Imaging Research. TMS is useful because it uses magnetic fields to raise and lower the threshold for neural activity and might be able to alter this balance between excitation and inhibition. With EEG, we're measuring spontaneous neural activity or brain waves and using those patterns to activity to test whether we can infer the balance between excitation and inhibition. Again, these are part of a broad suite of cutting-edge tools we have available to us here at the Center for Advanced Human Brain Imaging Research. In my own work, I also use fMRI, or functional magnetic resonance imaging, to measure changes in brain activity at fine spatial scales while people are at rest and while they're performing tasks. We also use eye tracking and pupillometry as sensitive measures of behavior. And finally, we use high-performance computing to build models of brain dynamics and behavior. These tools drive fundamental discoveries to help us understand both basic brain function, but also what goes wrong in neurological and psychiatric disorders, and ultimately how to treat them.